Thank you very much. Dear colleagues, in my presentation, I would like to talk about the main areas of the research in our laboratory of soil carbon and microbial ecology. So it will be a kind of review presentation, review of our research. And currently, I would like to highlight four primary research areas in which our lab is working now, and these four areas actually are really interconnected between each other. And the first one is soil metagenomics and the research on soil microbial diversity, which includes studies on microbial composition and abundance in soil, intermicrobial networks, also microbial communities of soil niches such as, for instance, soil rhizosphere, and finally the effects of different environmental factors on soil microbial community and abundance. The second one is research on soil carbon sequestration issues, which are connected to determination of soil carbon pools, such as particulate organic matter, mineral-associated organic matter, microbial biomass carbon, and also our colleagues from our lab determine soil organic matter chemical structure, but different methods such as pyrolysis. And finally, we determine microbial kinetics and ecological strategies under different land use types. And the goal of these studies is to find the relationships between microbial parameters of soils and soil organic matter parameters, which can be related, and to reveal microbial drivers which control the processes of processes of carbon sequestration and mineralization. The third direction is related to soil fertility and plant health, and it includes the studies on rhizosphere microbiome and soil fertilization, the questions on plant nutrition and health, molecular assessment of soil phytopathogens in soil, and finally managing soil microbiome by adding substrates. And the last one is the microbial indicators and soil monitoring, because we're also aiming to find new microbiological indicators of soil ecological functions and try to perform the agroecological assessment of soil health using microbial indicators. Of course, that all of these issues are related to the problem of managing the soil microbiome, and it's really important because soil microbiome is actually the catalyst for advancing biotechnologies, and there are two strategies for managing the soil microbiome. The first one is based on the concept of prebiotics, and the second one is related to probiotics. And I believe that two of these strategies are really important and should be combined together and used together to reveal really good results and to develop new agrobiotechnologies, because it's important to use natural soil microbiota management through alteration of soil ecological conditions, and also introducing microorganisms into the soil and other niches like rhizosphere via inoculants. Well, and here I would like to tell, I cannot change the slide, I don't know why. Oh, thank you. Yes, and for this purpose, we use several long-term fertilizer experiments, which allows us to study the relationships between the chemical properties or organic matter and also microbial functions such as microbial diversity and the abundance of phytopathogens in soil and rhizosphere. We use long-term field experiments, long-term microplot experiments, and also we launched a new long-term field experiment in the region with different, with application of different composts. And here I would like to tell on one of our experiments, on one of our research, which is related to the effect of different fertilizer systems on soil and rhizosphere microbiomes. We selected 3 plant species: potato, white mustard, and maize, 2 fertilization types, mineral and organic. And we studied 2 niches: bulk soil without roots and rhizosphere soils at 2 stages of plant development. And we studied soil microbial biomass, the abundance of different microbes by real-time PCR, and the structure of microbial communities using DNA and RNA next-generation sequencing. So we had a lot of treatments, a lot of data, and we sequenced a lot of DNA and RNA to reveal the structure of active and total soil microbial communities. And we found that all of them mostly make 2 clusters which are controlled the type of fertilization. So the cluster of microbiomes under the effect of manure, and the other one is a cluster of microbiomes of soil and rhizosphere under the effect of NPK. And also we found that the microbial diversity strongly decreased after the application of high doses of NPK, and conversely increased after the application of organic fertilizers. So the high doses of fertilizers had a more significant effect on rhizosphere prokaryotic communities than the plants, and the application of organic fertilizers could restore prokaryotic diversity in the rhizosphere. And why it happens? Maybe someone can think that the increased diversity under application of organic fertilizers may be due to the introduction of some microbes. So we have tested this hypothesis and analyzed prokaryotic and fungal communities of manure microbiome, which was used in this experiment, and we found a lot of intestine microflora and also many fungi. But we found that all these microbes, mostly the microbes from manure microbiome, didn't found in soil samples, and altogether the number of survived manure microbes were strongly decreased with time, and even after 2 weeks, the number of detected genera from manure microbiome were almost even lower than 50 compared to 200 genera at the beginning. So as you can see, the survival of manure microbes in soil is really low and continued to decrease. But manure microbiome activated many minor soil taxa which didn't detect in soil without organic fertilizers. So that was a reason of the higher diversity in soil with organic fertilizers. Also, we performed the same study for rhizosphere and soil fungal communities under different fertilizer regimes, and we saw quite similar picture also again to clusters which are controlled by the fertilizer types. So we can conclude that higher doses of fertilizers governed not only prokaryotic but also fungal communities in the rhizosphere of agricultural plants. And this is interesting because it opens the possibility of manipulating native rhizosphere prokaryotic and fungal communities of agricultural plants by adding various types of substrates into soil to achieve improvements in soil microbial functions and enhance crop production. Also, we analyzed better diversity of our rhizosphere and soil microbiomes. Again, we found these clusters that bare fallow communities went to opposite directions. So we found that all our communities performed clusters dependent on fertilizer types. And also we tested the abundance of microbes, of fungi, bacteria, of archaea using a real-time PCR, and found that fertilizer was also the main contributor to the microbial abundance, and surpassed plant-related factors even in the abundance of microbes. Also, we tested microbial physiological activity under different fertilizer regimes, and as we found before, that mineral fertilizers decreased microbial abundance in soil, and it led to the decrease also in mean physiological activity of microbes in soil, both in the soil and rhizosphere. Why was it happened? We analyzed microbial community structure using next generation sequence and found that the abundance of nitrifiers and denitrifiers such as Nitrosatale and so on, and other nitrifying and denitrifying species, and the proportion of these microbes in microbial communities dramatically increased under application of NPK. So we found that a real huge amount of microbes in soil after long-term application of high doses of NPK were just several microbial genera with quite with the relation to a nitrogen cycle, not organic matter and not carbon cycle. So we can conclude that long-term application of increased doses of mineral fertilizers reduces the number of prokaryotic taxa, taxonomic diversity, and even functional activity in relation to carbon substrates into bulk soil and the rhizosphere. Also, many soil microbes are also soil-borne phytopathogens of agricultural plants such as Fusarium, Verticillium, Rhizoctonia, and some of our next presentations would be about this problem, and they talk about them. So it's important to develop molecular genetic approaches to detect and to quantify the number of phytopathogens in soil. And here is a we continue to analyze fungal communities in the rhizosphere and the soil under mineral and organic fertilizers. And as you remember, it was a decrease in microbial abundance and diversity after application of mineral fertilizers. And we picked the fungal taxa which are potential phytopathogens of agricultural crops, and we found that the long-term application of high doses of NPK also resulted in the accumulation and increase of potentially pathogenic fungi as well. And conversely, the application of manure decreased the abundance of most fungal phytopathogens. So we can conclude that soil ecological conditions have a significant impact on abundance of phytopathogens in soil. And also we try to develop the approaches to quantify the abundance of microbes, for instance, the abundance of Verticillium taxa in the rhizosphere and soil. And we found that, for instance, in some soils, the abundance of Verticillium alboatrum and Verticillium dahlia can be really low, even not detected, but in the rhizosphere, simultaneously, the abundance of these species could be really high. So it's really important direction of the research to quantify the abundance of phytopathogens in soil, and it would help to test biocontrol agents, the effectiveness of biocontrol agents in soil. Also we try to develop soil metagenomics approaches together with our colleagues from All Russian Institute of Agricultural Microbiology, and particularly with Professor Evgeny Andronov, and together with our colleagues from this institute, we try to work with full soil metagenomes, and for this purpose we collected five typical Russian soil types from natural reserves. And now we are studying on this project and try to find the differences in the genes and functional genes and metabolic pathways between these soils in different regions of Russia. And as I told before, the really important part of our laboratory research are related to soil carbon sequestration issues. So now we start actively working with this, and we make these studies with the support of state assignment of the Ministry of Education and Science, and the project, the study of microbial drivers of sequestration and storage of organic carbon in soils of agro-ecosystems. And we use many microbiological properties of soil which characterize soil microbiomes, such as taxonomic composition, abundance, microbial physiology like growth rates, substrate consumption rates, and also microbial indexes. And the soil properties which are related to soil organic matter, such as different pools of organic matter, molecular composition of organics in soil. And we try to relate these characteristics and to find which microbial properties can be real microbial drivers of the carbon organic matter accumulation in soil. And for instance, we tested one soil type, but under 4 types of land use, such as perennials, abandoned land, arable land, and pasture. And although these soils were in the one region and were located really closely to each other, but the organic carbon content was really contrast in these 4 soil types. But if we talk about microbial biomass carbon, it was characterized by a similar trend in distribution across different soil types. But if we, for example, if we when we tested bacterial abundance by real-time PCR or average multi-substrate activity, we found that these microbial properties had a completely different trend, with the highest values under the abandoned land, not under perennials. But also some of our microbial properties of soils, which are really important, but they didn't differ between different land use types. So, for instance, microbial growth rate of microbial communities was completely similar and didn't significantly differ between different land use types. But we just started this project, so I hope that maybe in the end of the year we would obtain all the results on this project. So what conclusions we can make? Fertilizers have a significant impact on both the soil and the rhizosphere microbiomes. long application of high doses of NPK reduces the diversity and of microbes, which may have negative consequences for plant health. And high doses of fertilizers not only govern prokaryotic communities, but also fungal communities in the rhizosphere. And this opens the possibility of manipulating native rhizosphere communities by long-term addition of various types of organic substrates into the soil. And finally, soil, climatic, and agrogenic factors significantly affect the composition, activity, and the functions of the microbiome in the soil and the rhizosphere. And the survival rate of introduced microorganisms is usually low and is determined by a wide range of biotic and abiotic factors. Thus, microbiological solutions in the field of plant growth and plant protection should combine the concepts of the management of the native soil microbiome and microbial inoculants. And finally, I want to thank the Russian Science Foundation and also Ministry of Science and Higher Education of the Russian Federation for their support of this project. Thank you for your attention, and if you have questions, I can ask for them. Thank you.